My name is Oluwase Yolawale. I'm the founder of GENOMAC HUB. I'm the host speaker. So we're going to be looking at an NGS comparative session where we'll be looking at comparative genomics and its applications to microbial, plant, and animal research. I repeat, we're looking at comparative genomics and its application to microbial, plant, and animal research. So what is genomics? This is a field of biology that focuses on the study of the complete genetic information of an organism, which includes the analysis of the structure, function, and evolution of all the genes and the interactions within an organism's DNA. So this is just the definition of genomics. Like I said, it's an aspect of biology that is focused on the study of genetic information. Now, when you hear about genetic information, what's the first thing that comes to your mind? That should be DNA, all right, or RNA. And also you can talk about genetic information to be relating to proteomics. That's when you're talking about protein. So it means that genomics is an aspect of exploring the genetic information, which is DNA, RNA of an organism. And this particular genomics aspect of genomics covers the sequencing of DNA samples down to analysis of the structure and the function, and as well as the evolution of these genes and their interactions within an organism and across organisms. That means across different species, either if you're working with plant, animal, or microbial organisms. All right. So that is genomics for you. So we have the DNA and the flow of information as far as genomics is concerned. I repeat myself, we have the DNA and the flow of this genetic information in the living organisms, all right? And we understand that DNA is the genetic material that encodes the information necessary for an organism to develop and function, all right? DNA is the genetic material that encodes the information necessary for an organism to develop and function. So I would like to say in this way that everything that made up an organism is actually encoded by a gene. All right, everything that encodes a plant, everything that encodes a microorganism, be it bacteria, be it fungi, be it virus or archaea, all right, they are encoded, all these things, either the structural, the functional, the genotype, the phenotype, the morphological or the physiological, right? They are encoded by a specific a certain genes. So there is a flow of information from the very source of the organisms, the very source of the this information, all right, which is DNA. So here we have the double-stranded, with complementary strand, which is the AT and CG, right? That is adenine, thymine, cytosine, and guanine. All right. Now this is the 4 nucleotide bases, all right, that build up. That is the basic, building structure of the building block of DNA. All right. So if you're talking about DNA, simply the A, the T, and the C, and the G. All right. And this is what we, what we see as DNA. All right. This is what we see as as DNA. So now, there is certain gaps. There's a specific gaps when it's relating to talking about how DNA, is being transformed, is being transformed to protein. All right. So we have the DNA, the RNA, and the protein. So this is like the practical flow of genetic information. And we understand that we have 2 categories of organisms, right? We have the prokaryotic and then we have the eukaryotic, all right? The prokaryotic, where you're talking about microorganisms, and the eukaryotic, where you're talking about the plant, the animal, right? And the likes, okay? So, and we understand that DNA is encoded in the nucleus as far as eukaryotic is concerned, but prokaryotic does not have nucleus, you know that, right? So we can say that there is a flow of information from DNA, RNA, and protein. And for DNA to become an RNA, all right, it's actually been, we call that transcription. Why RNA, move into protein, right? As you can see on my screen, is what? that is what we call translation. All right, translation. So basically, the art of the flow of genetic information in an organism is like from the DNA being transcribed to the RNA, and RNA being translated to the protein. All right. So we are still going to explore more because we don't have time. Let me just continue. So we have what we call codons and translations. You know, I was talking about DNA, and probably many of you will wonder what DNA is all about. All right, you may be wondering what DNA is majorly about. All right, so let me just show you a practical illustrations of what a DNA of a particular gene or an organism is all about. Okay, so I'm going to be sharing a particular software which I'm going to talk to you about in the long run. All right, as we proceed. All right, I'm going to talk to you about this particular software. So some of you may be aware about that, those who joined us last week. So this is Genome software, but I just want to explore a DNA of a bacteria, which is Lactobacillus fermentum. Okay, this is Lactobacillus fermentum. All right, I hope you can see my screen. So I just clicked here. All right, an organism which is Lactobacillus fermentum or Lactobacillus fermentum. This is just the other name. All right, the name has changed. All right, the genus name has changed, the generic name. All right, so this is the old genome. All right, this is the old genome of this organism, which is a bacteria. And as you can see here, I just clicked here, and then the information of this particular genome is showing here. So what you see here are nucleotide bases, which is the 4 nucleotide bases that made up the DNA. So what I'm actually viewing here, I'm looking at the entire DNA sequence of this organism. All right, the entire DNA sequence of this organism. Can you see? This is Genome software. I'm going to share with you guys the link to download the software if you haven't downloaded the software. All right, it's a genetic software, though I'm still going to talk to you about the software in the long run as far as this workshop is concerned. All right, so this is the DNA. As you can see, let me just try to zoom this DNA. All right, I hope you can see it. Let me wrap it. I just wrap it now so you can see what I'm saying. Let me remove the annotation. Now you can see the DNA. All right, now you can see the DNA. So let me do something very quickly so that you can see what I'm just trying to do. Please mute yourself if you know you are muted. So I just did a stylized DNA helix. I just clicked the stylized DNA helix. All right, so this is stylized DNA helix, and this DNA helix is talking about the double-stranded DNA structure. All right, the helical structure of DNA. And here you can see the T, this is like TTT, TAACGAA. So this is like the DNA sequence. Okay. Now I was talking to you about on my slide. I was showing you the codons. All right, I was showing you the codons. I was showing you the codons and and the translation. So as you can see here, we have the UUU, UUC, UUA, and the UUG. All right. So this is taught to make you understand that we have 3 nucleotide bases that code for a codon. All right. 3 nucleotide bases that made a codon, and this codon encodes an amino acid. So previously I was telling you that DNA transcribed to RNA and then RNA transcribed to protein. So you see this ACGT is the DNA, but when the T is changing to U, which is timing changing to uracil, that is where the transcription takes place. That is to show you that transcription has taken place, and then we have the RNA. But you see, before this RNA can become protein, there must be what we call codon, which is what I'm trying to explain to you. All right, this which is what I'm trying to explain to you. So 3 nucleotide bases made up a codon that codes for an amino acid, and these are the essential amino acids that we have, and these are the codon that code for it. Now back to the software. All right, let's go back to the software. Going back to the software, Going back to the software, you get to discover that we have the double-stranded helix, and you can see the green here. The green here shows that everything here is the T, because we have, you know, the colors of this ACGT, the A, the C, the G, and the T. All right. So the T is the green, and you can see the helix here, and then we have the other red one, which is the A. So it means that A binds with T, or T binds with A, and G binds with C. All right. So let me remove this helical structure. Let me remove the helical structure, and then show you. Let me translate. Let me translate for you. So right now I've translated. Should be translating. All right. It's being translated. So as you can see, we have the TTT. So the T here is coding for F as an amino acid, abbreviation of an amino acid, and that F is what? Phenylalanine. Can you see it now? So it means this is how the flow information occurs. All right. So this is just a basic introduction, introducing you to the genomics aspect of what we are about to do. All right, about to engage. Now we have a genomic research is aimed at DNA and RNA sequencing. All right, and it's also aimed at analyzing the structure and function of the DNA, identifying the genes and their function. All right, identifying what? The genes and their function, identifying the identifying gene expression and the regulation, analyzing genetic variations and mutations. Then we get to understand that the genetic basis of trait and disease across species, and developing new treatments and therapies based on this knowledge. So it means that the essence of genomics, it is that it gives us understanding to you know, in knowing the genetic basis of certain traits, either in plants, on animal, or probably microorganisms. And when we talk about disease across species, like, you know, animals, or, you know, the humans, and, you know, probably the poultry and the others, and also development of treatment and therapy. So genomics has been able to enable these areas of research. Then we have, what is comparative genomics now? So this is just the previous things I've talked about is just the aspect of genomics, introducing you to genomics. Now we have comparative genomics. So basically, what is comparative genomics? This is the field of biology research in which the genome sequences of different organisms are compared. I repeat myself. Comparative genomics is an aspect of a field of biology, all right, that involves genome sequences, just like what I showed you from the genetic software, which I'm going to be sharing the link to you on how to download that particular software, all right? So is an aspect of biological research, all right, that engage these sequences, either the sequence of a particular gene or sequence of the entire structure of a particular organism. And then you compare the sequence. And this sequence could be from humans, could be from mouse, or variety of different organisms, from bacteria to chimpanzees and the rest. So, and it could be a comparison between the same or different species. All right, so you can be doing a master's study or a PhD study where you need to explore an aspect of comparative genomics. Listen, let me tell you a secret. You cannot actually fully explore the aspect of genomics of bioinformatics without doing comparison. All right, I repeat myself. You cannot do or explore bioinformatics, all right, without fully engaging, I repeat myself, without fully engaging the comparative aspect of what you are doing. So it means, hello, please, are you following? Please, can you reply the chat room if you can hear me?
Yes.
Yes, we can hear you.
Amazing, amazing. Thank you so much. All right, all right. So I'm going to tell you the software. Somebody's asking what the software is. Let's just take it step by step. I really want you to understand this. Please, can you All right, I think, yeah. I was saying previously that you cannot do or explore comparative genomics. I mean, you cannot explore genomics without actually engaging comparative genomics. For every genomics study that you do, genomics research, as either in your undergraduate, master's or PhD, or maybe in your research place or your research institutes or wherever you're working, you cannot do genomics without doing comparative genomics. You know, one of the reasons why we have 1,000 genomics projects that took place in UK was because of comparative genomics. And this 1,000 genomics was actually a kind of project to explore the genomics, 1,000 genomes of humans. So the project was about sequencing 1,000 genomes of 1,000 humans. All right. And, you know, the idea about that is that scientists wants to do a kind of comparative genomics study or research on different humans, different patients, with different kind of diseases, to understand evolutions, to understand emergence, to understand diversity, to understand differences that occur, across species. Why? Because by the time you are doing a comparative study, you'll be able to discover certain areas that is kind of a bit different, between a patient and the others. Now, let me give you an instance. If you get a cancer genomics, a cancer DNA sequence, all right, and you get a cancer DNA sequence of an early stage cancer patient, and you get the cancer DNA sequence of late stage cancer patient, probably let's say it's actually breast cancer, right? Breast cancer, and there are two kind of people, or let's just say three kind of people. All right, the normal breast DNA sequence, all right, you understand, your DNA sequence of a particular gene from the breast of a normal person, and you get that of someone who just start developing lumps, like lumps in the breast, and then you get that one of somebody who is experiencing a late stage breast cancer. Now, let me tell you something. For you to understand what is going on, what has took place, or what has happened, the difference in mutations, all right, difference in the expressions of this gene, you cannot do without comparative genomics. You must do comparative genomics to understand, okay, what is the difference, or what is actually what aspect of DNA sequence made up a normal person, you understand, without cancer, and the person that has early stage cancer and the person that has late stage cancer. Now, let me give you a scenario. It is probably that there will not be any DNA mutation in the normal person that has not that does not have breast cancer, and then it is likely that you have certain numbers of mutations in the early stage cancer person, right? And then you have a very much diverse mutation, right? Like, so you will definitely be seeing differences. And then by the time you do this analysis, you study the genes, you study the genome, you'll be able to come up with an innovative information, all right? A kind of information that is insightful, that is very, very important in order to probably come up with an innovative solution, all right? Come up with therapeutic measures, all right? Or probably understand these expressions of this gene and be able to target a specific region of mutation that you can actually develop a drug that can target that particular point, that can target to that particular point, maybe for certain corrections and some of these things. I hope that is very, very clear. Is that understood? Can I see an answer the chat room? Is it very clear? Can I just answer the chat room? Okay, you don't need to reply with your sound, just answer the chat room with a yes. Is it very clear? Amazing. Thank you so much. Mayowa, Hamed, Zulfa, thank you. Bisma, Razi, thank you. Magana, thank you. Babalaye, Jayashri. Thank you guys. Thank you guys. Amazing. Amazing. It shows that you guys are following. It shows that you guys are following. So this is the idea. This is the idea of comparative genomics. Just understand this, and then every other aspect of genomics that you engage or you embarked on, I tell you this. You will definitely, be able to advance and be successful. You will understand. You will advance and you'll be successful. Because that is it. That is it. All right. So let's proceed. Research applications of comparative genomics. Research applications of comparative genomics. All right, I think I'm back. So to some of you who have interest in microbial studies, all right, and probably also you're interested in microbiology, food, and all of these things, and then maybe you're interested in plant viral disease and all of this. So this is an aspect that you would love to explore. All right, don't forget, you just need to wait with us for a bit longer because I'm going to be sharing with you a hands-on practical session of comparative genomics with this software, and I'm going to be sharing with you the link to download this software to use a trial version of it. All right, to use a trial version of it. All right, so comparative genomics is used to understand the evolution and diversity of bacterial and viral pathogens that cause disease in humans and animals. This information can be used to develop new treatments and vaccines to combat these pathogens. Do you understand that? I think that's one of the reasons why UK did 1,000 genome project on humans. All right, I think that's one of the reasons, because they wanted to do a vast comparative measures to see what is actually taking place, to come up with insightful data-driven a picture, all right, for certain decision in the research aspect, probably for drug or whatever therapeutic products or whatever they want to do. Now we have the comparative genomics is also used to study the evolution and ecology of microbial communities in different environments. Now this is aspect of metagenomics. Probably some of you are here, you are interested in metagenomics. All right, this is an aspect for you. It means that you cannot do metagenomics without comparative genomics. That is just it, because metagenomics is actually comparison. You'll be able to explore all these areas of interest, and then especially when you're talking about soil, water, and human gut. This can help you understand how microbes interact with each other and the environment, and how they contribute to important processes like nutrition cycle and carbon fixation. This is an aspect of metagenomics. I heard about the project in China where they were using metagenomics to study the growth of plants. I know you will say, how is microbe related to plant? Actually, it's very, very related. It's very, very, very related, especially when it comes to nutrition cycle and carbon fixation in the soil. All right, and carbon fixation in the soil. So it's very, very interesting. So we have the plant, which is comparative genomics, is used to study the evolution of plant genomes and the mechanism of plant adaptation to different environments. Can you understand? It's used in the evolution of plant genomes. This information can be used to develop new crops. New crops. Now, I'm going to share a practical session about these things, like what I'm trying to say here, like what I'm trying to make you understand, okay? This information can be used to what? To develop new crops that are more resistant to pests, disease, and environmental stressors. Now, let me give you a scenario. Imagine there's this plant, a maize plant let me use India. This particular maize plant has been known to develop resistance to droughts. So every other maize plant die because of there's lack of water in 2 months. All right, lack of rainfall in 2 months. But this particular maize plant is able to resist this drought. It's able to resist because it has a specific gene that is coding for that particular trait in the plant. You get my point? So it means that this particular plant has probably has developed a gene over time because of evolution. You understand? And this gene has occurred because of certain variations, good variations, let me put it that way, in the plant. And it has, and this thing has become a new trait. Now, what do you need to do? You need to do comparative genomics. Compare that plant in India with every other plant from anywhere. And then by the time you do that, since it's the same plant, the same species, or the same genus, they will have high percentage similarity. But there will be a place of difference, because comparative genomics is just about that, knowing the difference and similarity that occurs between organisms of the same species or different species and the likes. So you will then be able to locate that region, and then you'll be able to locate the gene of interest. Now, by the time you locate that area of interest, you study the area, the DNA sequence in that particular region, you study the gene, you understand the gene, and that is where you can now begin to come up with insight on how you can copy these genes or do some crossbreeding in the plant to make sure that other plants also have these features and all of these things. You get about it? So that is a great idea. That is where comparative genomics is very, very powerful. Then we have comparative genomics also used to study the genetic basis of plant traits. I already say that are important for agriculture, so that yield, nutrition contents. I was talking about stress, right? Tolerance, all right. But here we're talking about nutrition contents and yieldedness and yielding, right? This can also help us identify genes and genetic variations that can be used in breeding programs to improve crop performance and the likes. Then we have in animals as well. Comparative genomics is used to study the genetic basis of plant traits in animals, such as behavior, disease resistance, adaptation to different environments. This information can be used to develop new treatments and therapies for animal health, to improve breeding programs for livestock and the likes. Then we have the comparative genomics is used to study the evolution of animal genomes and the relationship between different species. Amazing. Amazing, amazing. This is insightful. This is insightful. And let me just proceed because of our time. The research applications of microbial genomics, all right? I'm then going to, we've already talked about the research applications, but here I just want to, I just want to introduce you to a practical research area, all right? So we have two of them. Number one is alignment, all right? Whole genome alignment or target gene alignment. All right, whole genome alignment and target gene alignment. All right, the other one we have pangenome and evolutionary study. All right, so like the first one is whole genome or targeted gene alignment. All right, to look for unique genes, practical unique genes. No certain genes that is not present in the other genomes and is present in your genome. Either you're working with plant, animal, or whatever kind of species you're working on. The other one is pangenome, and the other one we have evolutionary study. Now, we may not be able to explore all of this, but I'm going to show you a practical section as touching alignment. All right, as touching alignment. So I'm going to treat whole genome. The same concept also can also be used in the targeted gene or whatever you want to do. So let's dive into the practical session. All right. But before I go through, I'm going to share the slide. Before we dive into the practical session, please, guys, can you just indicate on the group? Just indicate on the group if the theoretical session, the explanations is very, very clear to you. At the chat room, please. At the chat room. Because we are diving into the practical session right away. Don't forget, whatever areas of study or field of study you are, in as much as you are interested in plant genomics, comparative genomics would be very, very, very amazing, all right, for you to explore. Very, very amazing for you guys to explore, all right? You can't do without it. You can't do without it. That's just the truth. You can't do without it. All right, so let's dive into this section. I can see you guys, your chat. Amazing. Thank you, guys. Thank you, guys. So let's dive into this section. Thank you. That's okay. All right, so let's dive into the. So the first thing I'm going to be introducing you guys to is what we call database. All right, we have genomics database, and this database are online servers. All right, online web servers that have been built for the storage of genomic sequences. All right, and even for carrying out certain kinds of analysis. So let's check NCBI database. Probably some of you have heard of it. If you have heard of NCBI database, can you reply with a yes? If you are aware of NCBI database, just reply with a yes at the chat room. And if not, you can reply with a no very quickly. NCBI database, do you know what that is? Have you heard about that? Okay, I can see a no. I can see a no as well. Amazing. Thank you for the honest answer. Thank you for the honest answer. So I'm going to be taking you through NCBI database. So just go to Google. Let me repeat it again. Go to Google and search NCBI database. All right, NCBI database. Search on Google for NCBI database. NCBI database. Amazing. Thank you for the honest questions. Never heard of it. Amazing. So just search for NCBI database. NCBI database. All right. So as you can see, I've searched for it, and this is it. So just click it, and it's going to take you to the web server directly. And here's the web server. You can just do that with your phone if you want. You can, if you have a laptop, whatever you have, you can just practice it. It's going to be amazing if we do it together. All right. So now let's proceed. Now we have the database. Now you can see all databases. This is the web server, and it has certain databases that have been built for specific reasons. All right, and here I'm going to click. I clicked here, and then we have our database. But we are looking for something. We're actually looking for something. All right. So you can. So there are two things, as I told you. There's a gene aspect, and there is the whole genome aspect. So if you want to do comparative genomics, you can work with gene. The gene is a targeted sequence, because targeted sequence is talking about a specific gene. Now this gene would be a specific trait. Then we have a whole genome, meaning the entire DNA sequence of the whole genome of an organism. Now let's proceed. To do that, let's just dive in. So let's work on the whole genome. Now to work on the whole genome, all right, to work on the whole genome, you can go to nucleotide.
Hello.
Click all database.
Frozen. The screen is frozen.
All right, let me share. So I'm sharing again. I believe you can see it now. You can reply with a yes at the chat room.
Yes.
Amazing, amazing.
Yeah,
so I just click all databases. You can see all databases.
Screen.
Hello? Please reply at the chat room. Thank you.
I cannot see the screen.
I believe others can see the screen, right? I believe others can see the screen.
Yes, we can see the screen.
Yes, sir, we can.
Yes.
Yes, we can. Yes, we can.
See the screen clear.
Yes, we can.
Yes, I can see the screen.
Yes, I can see
the screen. Please focus on your.
All right, so let's proceed. Click all databases. Right here, you will see all databases. So click it, scroll down and click nucleotide, nucleotide. And from there, can somebody give me an example of an organism we can work with? A whole genome of an organism we can work with. I would like bacteria because of our time. If we work with plants, it's gonna be huge.
SARS-CoV-2.
E. coli.
All right, let's work with E.
SARS-CoV-2.
coli. Amazing. Let's work with E. coli. Everybody is familiar with E. coli. All right, and let's search for E. coli. So let me say whole genome of E. coli. Whole genome of E. coli. And here I am viewing the whole genome of E. coli. By the time you scroll down, you get to discover that the sequence is kind of a bit small. But if you want to look at the whole genomes of E. coli, all right, you could just go to all genomes. You can click all genomes. So I just clicked all genomes. And it took me to the genomes. All right, it took me to the genomes, and these are the genomes that are available. All right, and those that submit them. This is just an example. This is a reference sequence. This is a reference sequence. All right, so now we need to download the gene bank. Now I will explain what gene bank is. Gene bank is the entire genome structure of E. coli or any other organism that has the annotated genes in them. Now the annotated genes in them, I'm talking about the entire DNA sequence of E. coli has already been annotated. It has been predicted. All right. And what was predicted are the genes and the proteins that are in it. I repeat myself, are the genes and what? The proteins that are in it. So what you just need to do basically is download the gene bank. So I'm going to download this reference sample because whenever you want to do comparative genomics, you need a reference sample. Now this reference sample could be your own sample if you are doing PhD or masters and you're working with whole genome. All right. So your own sequence, the one you did from the lab, it's the reference, is your own reference. So you will use that as the reference sequence. Is that clear? That would be your reference. But I don't have one, and I want to work with the reference sequence of NCBI.
Hello, sir. Hello, sir.
All right. Hello, who is that?
Yes, can you please?
Are we downloading the reference sequence?
Yes, we will. Please just follow me. All right. Just follow me. You understand everything I do so that we can avoid distraction. And we don't have a lot of time anymore. Okay, we don't have much time.
Can you please repeat this?
Download.
Because I didn't find what you found, actually. That's why. Can you repeat that very quick?
Okay, can I repeat the steps?
Yeah.
Yes, sure.
Amazing, amazing. So please kindly mute yourself so that we can avoid distractions. I would appreciate that, okay? So I'm going to repeat all over again. I'm going to repeat all over again. So search for NCBI Database on Google and click it just like I did. And then you come to the web server. On the web server, we have a lot of databases on it that is particular to a specific genomic information. But here we are interested in, you can either click nucleotide or you click the genome. All right, you can click the nucleotide or you click the genome. But let me work with the nucleotide, okay? Because the nucleotide is talking about the entire genome structure of the organism. Even the genome also is also the same. So I'm going to type Escherichia coli whole genome. You want to search it, all right? And click enter. Just search it out here. And then here it is, all right? Here it is. So we have quite a numbers of results here. Here we have, is it 12 million? Is it up to 12 million? Yeah, 12 million results on NCBI database. That's amazing, including the genes actually. All right, but I'm not interested in the other results. I'm just interested in all genomes here. So let me click on all genomes, right click on it, and open a new tab, which is right here. This is where we were before I repeat myself. All right. So now we have the genomes of E. coli. But here, the reason why I come to this point is that I was interested, or I am interested in the reference sequence. And I said that if you are a PhD student or master student or a researcher, and you want to do a comparative genomics of plant, animal, or a microbial genomes, and you're doing the same thing I'm doing right now to get the genomes of that particular organism you want to work on, all you just need to do is look for a reference sequence. Now, if you're a PhD and a master student and you're doing your program and you want to do comparative genomics, what you need to make as your reference, or what needs to become a reference for you, is your own sequence. Your own sequence. That means the sample you send to the lab for DNA sequencing, for NGS data sequencing. That means the sequencing in aspect where you get the entire DNA sequence of that particular organism or a specific gene of interest. Remember that's what I told you. There's two categories to this comparative genomes. It can have a gene or the whole genome. But I want to show you the whole genome because in the whole genome you will do the gene. All right. So now this is the reference sequence of NCBI. Whenever you see this sign, it shows reference sequence. So I want to download this because I don't have a DNA sequence for my study. I want to use this as my study. So I'm getting the DNA sequence from the NCBI. All right. Then I click here. Can you see these three dots? I clicked here. And while I do that,
the screen is frozen, Doctor.
Hello?
The screen is frozen again.
I think that should be your network. Everybody, is it the same with you?
No, it's
No,
perfect.
no.
You can see the screen. Screen is properly working.
It's clear
Amazing.
now.
Thank you.
It's
So you
clear.
can mute yourself now. You can mute now. Thank you. So
Can
please.
you do reshare? Because when you do the reshare, it's
We are definitely going to share the recording, okay? So please, let's avoid distraction. Let me finish this. We don't have much time. All right, we still have some things to do.
Please take your time. Please take your time. You're doing great.
Thank you so much. All right.
Hello, you are muted, sir. We can't hear you.
Are you there?
Hello, I'm back. I believe you can hear me. Just reply to the chat room, okay? All right. I believe you can hear me now. Very clear. Amazing. Thank you. So this is the information of that particular sequence. You know, we just clicked here to view the information of this reference sequence. And this is the statistical information. This is the genome sequence information, where we have the number of scaffold, the number of chromosomes in it, all right, the assembly level, and then, you know, it's complete, and then other things. So you can just study the coding regions, so that the proteins and the non-coding regions. All right. So, but this is just the details. But I want to download it, and I want to download the gene bank. That I was explaining the gene bank previously, and I told you the gene bank contains the entire DNA sequence of the organism, including the genes in there. The coding structures, I mean the coding sequences, the proteins in there. The functional proteins are what they code for in the organism. So this is E. coli, and then we are going to download the entire genome sequence, and then we also download the gene bank, which is the genome sequence. Now I don't want the genome sequence, I want the genome sequence and the annotation. Can you see it? I want the genome sequence and the annotation all together. So you don't need to click this, you just need to click this. Now there are two reference sequences. I could decide to download just one reference sequence or download all of the reference sequence. So let's work with just one of it, all right? Because I just need one of it, okay? And remember the options I pick is sequence and annotation. I scroll down and I click download. I scroll down and I click download. Now I'm downloading. I hope you can see that. You can reply with a yes at the chat room if you can see. Yeah, you can see that. Thank you. So it's downloading. As you can see, it's downloading. And the size is 6.5 and it has completely downloaded. Now I want to view that what I just downloaded, and this is it. So let me create a new folder. And I could call it E. coli comparative right? And I would then copy this into it and then open it and extract this. I will extract here and it's extracting, and I go to NCBI Database, I go to the data, and you know, I think it downloaded all the two reference, but I just need one of it. I think it still download both of them. So but I just need one of it. So I just need to click this, copy it, and go back to the you know, the folder and paste. All right. So and then I need to rename this to E. coli reference genome. All right. So I just name it E. coli reference genome. Now we need to go back. So we have the reference genome now. So imagine this is your reference genome. So you need to get other sequences, other genomes for comparative genomics. Remember, this is microbial. You can do the same for plant and animal. So I will download I can download every other ones. Like I just need to keep clicking. So I can click like 5 of it, depending on what I want to work with. And then come here and click download download package. All right, can you see? So I just need to do the so I just pick 5 of it. You can pick as many depending on your objective of your research. And then the sequence and annotation. So I just need to download that. All right, sequence and annotation. I just need to download that. You can do that one after the other, just like what I did here, and you can just, you know, pick randomly based on what you know, what's it called now? What this thing is all about. Sorry, the strain is all about. So I think it's fully downloaded. Let me go back, and here it is. All right. So let me create. Let me just cut it. All right. Take it to the folder and create another folder. So let me just say new folder, and I paste and extract. So it's extracting, and then it's done. So I need to go to the data. So this all the sequence I downloaded. So I just need them. I need to extract them. All right. I need to cut them. I need to cut them here. Open it. And make sure you save it with a strain. And you get a strain here. Can you see E. coli strain K12? Substrain this. All right. You close it. You rename it. You see? So you go back to the new folder and the data. You know, we already copied this. We already cut this. So we get this as well. We cut it. All right, and paste. You keep doing that. Please, can you mute? I believe you can hear me now. I think it's the same strain. What I just downloaded is still the same strain. All right, so let's go back. Not there. Here. So you can check. Let's just check it out. Is it still the same strain? 1655. Seems to be the same strain we downloaded. So I think it's better you do it one after the other. I hope that is clear. So you could just do it one after the other. I hope that is clear. So I'm downloading the other one. All right, so here, let's extract. Okay, I didn't want it to extract elsewhere, so I need to create a new folder. So here is it. Amazing. And this is substrate. Still the same substrate. Amazing. All right. So let's just copy it. Let's just use this as an example because of our time, and let's paste. So basically, you just have to download other sequences, but if you don't want to download other sequences from there, you can actually use this other previous one, E. coli whole genome, and then you keep searching. So let's check for 100 here, 500, like page 500. We're going to the page 500. Okay, and then we have, so you keep seeing, you know, the whole genome. You should see the whole genome strain here. All right. Let's use complete. I really want you to get this complete CDS. At times, it depends on how it's being saved on the database. Aha, yeah. So you use Escherichia coli complete genome. All right. Now, can you see the complete genome? So if you don't want to use this aspect, this area, so just get the reference sample and come here. So you can click the gene bank or you click the organism itself because these are different strains. So let's click the first one, which is this strain. You have it, it's quite a lot. You will know a complete genome by its numbers of sequence, okay? And go to just
Excuse me, you are muted.
I believe you can hear me now. So I'm deleting what. Yeah, amazing. I'm deleting what we don't need. I'm deleting what we don't need. So I think this is still the same as the K12 substrate MG1655. So this is a reference strain and this other two. Now the other ones are the subject strains. You can download as many as possible. Now just go to your this is what's it called now? This is Genius software. Now this Genius software can do a lot of analysis like transcriptome analysis, variant analysis, comparative genomics analysis, clinical genomics, plant genomics, and the likes. This software can do that. So we'll be using this software because it has a lot of plugins. Now, I will share with you the link to download this software. You can use the trial version, but it's paid. Now, I'm going to give you a gift at the end of this program for those that wait. All right, for those that will wait till the end of this workshop, I'm going to give you, well, I mean, the company is going to give you guys a gift. All right. And this gift will be to access the lifetime activation key of this software. Now, if you want to get this software in the market, all right, if you want to get this software, that means price for Genius software. Price for Genius software. Can you see? $200 per year for students. This is just per year. So it means if you want to be using this for every year, you just keep going to be paying $200 every year. So, but then we have the lifetime activation key, which we're going to give you guys. This activation key has been with us for quite some time because of our work with Genius software in regards to the use of the software by the company. All right. So, but we're going to give you as a gift. But I'm going to explain to you how the gift will be at the end of this workshop. So just stay with us until we're through, okay? So now, this is Genius software. This is where you upload, you create a folder. I'm going to show you how you can use it. Hello, please, can you mute? Hello, can you hear me? Reply with a yes at the chat room.
Yes.
Amazing. Yes. Amazing. Thank you. So now, the moment you activate, you download, you install this software in your system, use trial version first. Now, when you finish using trial version, you just go to help and activate license to be able to use the license. We're going to show you how you get the license, but I will send you the link to download the software. All right, on the WhatsApp group. We're going to do that.
Excuse me, you are muted again.
Please, by the time you install this software, make sure you go to tools, plugins, and make sure you are online. Some plugins will show forth here. Just keep clicking install until you finish installing everything, because if you don't install, you won't be able to use the software. All right, you need to install those plugins. Those plugins are individual tools for doing different kind of bioinformatics and genomics analysis. So now when you are done with that, click local, go to file and click new folder. And when you click new folder, you can name it comparative genomics, or whatever you want to name it. Click OK, and then you see it shows here. Click it, go back to file and go to import from file. Import, from file. So you are importing those gene banks you already downloaded. So let me go to my downloads, E. coli comparative project, and these are the sequences I want to upload. So I want to upload the sequences, so you can even do that one after the other, like import. So I'm importing right now. So it's creating the document and importing the DNA sequence and the annotation, which is the genes and the proteins. All right. All right, it's done. It's done. All right. And it's loading. It's done and it's loading. It may take some time to load.
So yeah, there's two options: create a list or keep sequence separate. So which one will select?
Create like keep sequence separate. Keep sequence separate since you are uploading everything. Okay, keep sequence separate. It's amazing you are following me. Amazing. So now this is the circular genome map of this particular reference sequence, and you can see a lot of things here. A lot of sequences are showing forth here. All right. So by the time I click this region here, as you can see this yellow icon, you will see the CDS. The CDS is coding region or coding sequence. You will see the numbers of genes, miscellaneous, mobile gene element, ncRNA, original replication, RNA source, and tRNA. So these are some of the informations, the genes, the features, genomic features, properties that made up the entire genome of this particular E. coli. And this is a circular view. So, but let me view the linear view. So to view the linear view, just go to sequence and click circular sequence, and then you'll be able to see the linear view. You can zoom here to see all this, to see the the nucleotide basis, and then you can click this home icon and wrap it, okay, to keep seeing, and then scroll down to keep seeing everything. So you can see all the genes, the coding sequence that is present in the reference sequence. All right, is that clear? Are you guys following? You can reply the chat room. Are you guys following? You don't have much time. Oh my God.
Yes, following.
Amazing. Thank you so much. Thank you so much. Now, I'm going to import, or let me just try to rename this. Let me rename this edit name, edit underscore ref to indicate that it's a reference sequence. Okay. Keep changes in viewer. Save. All right. Discard external changes. Yeah. So now I want to import still in the same folder. Make sure you click the folder. Go back to file. Come to import from file. All right. And pick the rest of the string. So if you have like 10, 20 of it, just import it. So this is what somebody was asking. After this, is going to ask you create sequence list or keep sequence separate. So let's wait on it to create the document. Now, can you see keep sequence separate or create sequence list? So keep sequence separate. That's the option we need. All right, so we just uploaded it, still in the circular view, but we need it in the linear view. So just go to choose or sequence rather, and click the circular view again. We want to view it in a linear format. All right. You save it, and it's saving. So just in case yours may not bring the linear format, all right, so you don't need to worry. You don't need to worry.
Is it free software?
It's not free software, but at the end of this program, we have a gift on how you can get the free software, okay? I mean, on how you can get the activation key. Right, trial version. But we have a package for you guys to get the software, the activation key, because the activation key costs $200 per year.
So let's say you're having a MacBook. So what's next? Do you also have an activation key for Mac or a different link for downloading?
I would suggest that you partition or look for a way you can install Windows software on your system, right? So that you can be able to install this. Though there's an option for Mac as well. Okay, there's an option for MacBook. Is that clear?
Sure. Thanks.
All right, so it's done. So now we need to do a comparative genomics, which I told you in the previous slide that would be done through alignment. I repeat myself, that would be done through whole genome alignment. So what we're doing now is whole genome alignment. So if you want to do targeted gene, it's still the same method. All right, still the same method. Now we need to click everything. As I've done now, just click here and it will pick everything. Just click here and it will click everything. And go to align and assemble. Go to align whole genomes. Align whole genomes. And I just click that. So let me repeat myself. So I just picked everything again. Align and assemble. Align whole genomes. So use last alignment. There are two categories of alignment here. Use last alignment. Now you need to pick the reference. Remember, this is our reference, all right? This was our reference, all right? So you need to pick a target sequence, which is the reference, which is your own sequence for the comparative study. And when you do that, you click OK. So this software is going to align it based on the principles of alignment, all right? Based on the principles of what? Of alignment. Based on the principles of alignment that was done, okay? This kind of alignment is done in a lot of comparative research papers, a lot of genomics research papers. It's been done. Probably by next month we'll explore other areas of alignment, all right? Other areas of other areas of comparative genomics, which is pangenome, all right? But that's by next month ending in the next what's it called now? Webinar series, all right, which is next month ending the last Saturday of April. So can you see it's in 50% now? So the more genome sequence you have, the longer it is, all right? The longer it is. Now it's going, it's about to finish. So the next thing you just need to do is look for a regions of uniqueness. Can you see it has finished aligning? And it's displaying, it's trying to display the result. Now, this is the result. Amazing. This is the result of two sequences aligned to the reference, two E. coli strains aligned to the reference E. coli that we got. So if you have your own sample, that will be your reference. And then you align it, and then you discover, okay, what is actually the difference between these two regions, between these two sequences? What's the difference? We've already told you the essence of doing this. All right, we've told you. So this is E. coli, and you can see two lines here. This is an alignment line, a comparative line. This line you see here. All right. These two lines, or every dot line, the red and the blue line, signifies regions of similarities between these two other strains to the reference. Remember, this is the reference. Let me try to drag here so that you can see what I'm saying. This is the reference, and these are the other two. So this region here is the region of this particular genome, and the same with this is the region of this particular genome. This is comparative genomics. We have compared them, we have aligned them based on maximum likelihood, based on pairwise and regions of similarity. Now, by the time you come here and click this percentage sign, you'll be able to see the length, the sequences, the identical sites, the pairwise identity. So the pairwise identity is telling us this is the statistical result of the alignment that was done, that these other two samples is 98.7% similar to the reference sequence. Is that clear? 98.7% identical to the reference sequence. So it means, what is the, can somebody reply in the chat room? What is the percentage difference? Can somebody calculate the percentage difference for us? Can somebody calculate the percentage difference for us? That will be 2 point what?
1.3.
Amazing. So the difference between these other two samples to the reference sample is 2.3. And this difference is based on the reference compared to the what? Or the two compared to the reference. But actually the focus is on the reference, because the reference is yours. You get my point? It's 2.3, not 1.3. Is it 1.3? Really? Yeah? 98? Yeah, that's true. Yeah, that's amazing. That's 1.3. Yeah, 1.3. So now, these regions you see here, one is reverse and one is forward. Like, so this, wherever you see the blue is the forward, and then the red is just the reverse similarities. Okay. So then we have the identical sites. This is the total identical sites, and this is the sequences, and this is the length of the sequences. Okay. So now let's see these sequences here. I think it signifies the genes. So when you click here, you will see the genes, the CDS, the genes, the miscellaneous, and the likes. So by the time you zoom, you'll be able to see the genes. All right, you'll be able to see the genes. So let's like drag back here so that we can start from the beginning, like 1. So we can start from here. Now, how do you know what is unique to your reference genome compared to these other strains? Remember, the reference is the focus. So if you have a sequence, you have a target sequence in mind, that's your focus. That's what you're trying to see, what is unique in that reference. So if I'm working with a genome of a particular maize from India, so what am I doing? That would be the one from India that has developed a resistant gene, all right? A drought resistant gene, right? That would be my reference sequence, and that would be my target. So I want to look for regions of uniqueness that is found in my reference sequence and is not found in others, or regions that is found in mine and is found in the others, or that is found in one of it. Now, as you can see, this is a particular region and this is a particular gene. The gene is THRL gene, which is this THRL gene. And this particular gene, this is the coding sequence that is coding for this gene. And this particular gene is not found in this particular genome, but it's found in this particular genome. Why? Because we see this red sign. I hope that is clear. It's not found in this region, but it's found in this region. Now let's close in. So all these regions, all this gene you are seeing here, as far as this line is not there. Okay, now we see this blue line. So this is telling us that this particular gene is also found in this and is found in this. But we are looking for unique region. Unique region is where you will not find any line here and you will not find any line here. So it means that you have a gene here, but it's not present here. So this line is not showing up, which indicates that it is not present. Is that clear? So let's look for a unique area. Now, this is a unique area. Can you see? So this is telling us that this particular gene is present in this reference sequence, but it's not present in these other two sequences. Now, by the time you keep going, you keep seeing unique genes like that. Specific genes that is present in the reference, which is your own subject reference sample. You understand? Targeted sample, but it's not present in the others. Is that clear? Right? I hope that is clear. It's not present in the others. That is the mind. So now, by the time you discover that, oh, this gene is present in the sequence and it's not present here, it shows that it's unique to the reference. It's unique to the reference, and the reason why it's unique to the reference is because this particular reference genome probably has developed mutation over time. It has developed a kind of evolution, emergence over time. That is, it has new genes as it developed, has come up, right, in the genome, making it to look a bit different, or in terms of functionality based, different from the other genes. And now imagine this is a maize plant, and this is a gene that codes for drought resistance, and you get to discover that, okay, this gene is not found here. You get my point? This gene is not found here. I mean, it's found in the reference sample, it's not found in the other two. So that shows that, oh, this is a unique genome, and this is a gene of interest. This is a gene of interest to focus on. Now let's see. I just copied this gene. Now, how do you get more information about this gene? Just go to Google, because we're about to round up the the workshop session. So just search So what does the tax gene does? It's a charging product. Is a positive regulator of transcription from the viral. I think okay, what we're working actually on is T-tax gene. Provide a unique query environment for genes. All right. So this is T-tax gene product. So this is how you go. You just come to you just come to Google to make a vast research on that. Another thing you could do right now is that ChatGPT is out. ChatGPT is out. All right, ChatGPT is out. You could just like ask ChatGPT. You could ask ChatGPT what that gene does. Can we check ChatGPT? So you know you can know what the gene does. By the time you know what this particular gene does, then you now make an extensive research on that. Why is it here? You understand? You can relate it to the morphological or physiological function of this. And what is this gene? What protein is this gene coding for? It's still so the protein it's coding for. Here we find the translation and there's some other information that you need to discuss. Please, is that very clear, guys? I know you guys have a question. We're going to have time for questions. Don't forget, at the end of this workshop, we have a kind of a gift for you guys. We are motivated and inspired by you guys joining us. It's amazing. It's amazing. So now let me ask the role of this. All right, ChatGPT could not find information. All right, so there are numerous genes that have been named with the prefix TFA. All right. So in we stand for transcription factor. Okay, I think Google is also right, because at Google it was telling us about a kind of transcription. It is a positive regulator for transcription from the viral FTRA. So it means that this gene code for a transcription factor in E. coli. Is that clear? Okay, more information about the specific organism. So what is the role of that gene in E. coli? So let's see what it had to say. In E. coli, it has encodes a protein called transcription factor A, which is very correct, which is also known as alpha subunit of RNA polymerase. TFA is a crucial component of RNA polymerase haloenzyme, which is responsible for initiating transcription by binding to promotion regions of DNA. So you can do an explosive research by talking about this particular gene in E. coli, and then look for related papers that talks about it on Google. All right, look for related papers that talks about it in Google. Is that clear? Is that very, very clear? So that is it basically. That is it very basically. All right. So this is time for question. This time for question. So I would like to take 1 or 2 questions. I would like to take 1 or 2 questions. Then I'll introduce you guys to the package, gift package that we have for you guys for joining us today. So I would like to take a question. Go on, please. You can ask, you can also drop a question at the chat room, guys. You can drop a question at the chat room.
I want to take the first chance.
Okay, go on, please.
So I wonder what kind of files that I have to upload in Genius. Is it a GenBank file, the annotated one? If that's the case, so can you see?
Can you see? It's the GenBank file. GBFF file. GBFF. So the process, we are going to provide the recording. So you just follow the recording to know how it's been done. All right.
So it means that I have to do kind of annotation before uploading for the comparative genome analysis.
Yes, GenBank from NCBI Database. Okay.
No, I mean for if I have my own data.
So
okay. So I have to do an annotation and then proceed for comparative.
Have you done annotation?
No, because to get that GenBank file, I have to do kind of annotation.
Definitely, if to get the GenBank file of your sample, you have to do annotation. Definitely, you have to do that. And to do annotation, you can work with Procar. You can work with Procar. Just search for Procar or NCBI annotation, web server. Then you submit your sample. Once it's done, you download the GenBank, and then you do this comparative. Okay. So, but hello, are you with me?
Yeah, I'm following.
So if you want to know how to do the genome annotation, just wait. At the end of this workshop, I'm going to be introducing you to some packages that we have as a gift for you guys, and then we're going to be using that to, you can actually use that because we're going to be introducing you to functional genomics. So you can use functional genomics to annotate your genome, get the GenBank, and then do this. All right.
Awesome, excellent. We are waiting for that. Thank you.
All right. All right.
Please, how to identify a particular gene in different genome?
A particular gene in different genome. All right. So like I said, this is a gene, right?
Yes.
And the reason why you see this gene, the reason why you cannot see it here is because it's unique to this preference sequence. Now, for you to know which gene is found in other this thing is just for you to look for regions of similarity. Remember, I told you the regions. One of the regions of similarity is this line. How you identify that this particular gene is also present in this genome and in this other genome, because we just aligned. This is a genome and this is genome, and this is the region of the genome. This is another region. This is a region of this region. This genome. This region is specific to this. Now, this red sign is showing that this gene is also present here, but it's not present here. So this gene is present in the reference sequence, and it's telling you that this gene in the reference sequence is not present here, it's present here. You get my point?
Okay.
Is that clear? Now look at this gene. Look at this other gene. This particular gene is telling you that this gene is present here in this genome, and it's also present in this genome, because we can see this blue sign and this red sign. I told you the blue sign and the red sign is showing, indicating that this is a region of similarity compared to the regions of the reference. Is that very clear?
Yes, thank you.
Amazing, amazing.
Sorry, sir. Sorry, sir. Is it that it is not present there, or is not annotated in that particular reference? That's why it's not showing.
No. It's not that it's not annotated. All right. It is that it is not present. It is unique to the reference.
Okay.
Do you understand? Because the same DNA sequence in these two I mean, the DNA sequence between these two is also present in reference. And that is why it's unique. Is that clear? Because these regions are also annotated. Is that clear?
Okay, thank you.
So if it is present, it will show it. So, and that is why you do comparative genomics to look for unique genes. Genes that are unique to the reference are not found in the others. That's what it means.
Thank you.
Amazing. So because that is bioinformatics analysis for you, and this is comparative genomics, an aspect of it. So probably in the next, cohort, I mean, in the next webinar series, we explore the rest of the which is pangenome for the comparative genomics in plant, animal, or whatever you want to. Okay.
Hello, sir. Can you hear me, sir?
Yes, go on.
Okay, thank you, sir, for this beautiful lecture. Please, does it mean that the reference gene, is it the gene? Like if I'm working with a particular sample, so is it the sample I got from the lab that is my reference gene, or the one I downloaded? I don't really understand. Can you clarify?
The one you get from the reference, I mean the one you get from the lab, your own sample.
Okay, okay, all right, thank you, sir.
It's your own sample,
Okay, thank you, sir.
We have a question at the chat room. Please, sir, does it mean if I have a target sequence? We have a question at the chat room. I'm reading it out. Please, sir, does it mean if I have a target sequence and I want to find out if they are resistant conferring mutation, my sequence will rather be this reference sequence or rather the reference to it? Okay, I think I just answered that right now. So your sequence should be the reference sequence, okay, not the strain. Okay, it should be the reference.
Okay, thank you very much. Good evening.
Good evening. Another thing is this. I want to answer it again. See, please. There are two ways to this. If you want to look for unique gene in yours, all right, you must have to annotate yours before. But if you haven't annotate, you can get a reference annotation, and your sequence should now be. That means if you haven't annotated your gene, your sequence should not be this the other ones above. All right, it should be the reference. I mean the subject sequence. These are subject sequence. These are other strains. So your sample should be the other strains. Why? You have a reference sequence that you download from NCBI Database that is already annotated. So you understand that whatever is in the reference sequence is also present in yours. And if it's not present in the reference sequence, it's not present in yours. Is that clear? So it's vice versa. It depends on the objective of your research. Thank you.
Okay, sir. Thank you, sir. Please, I want to ask the position of the red or blue line. Does it matter? Because I think
it matters. It matters. It tells you that, oh, this thing that is found here is also found here or here. Now, don't mind the red or the blue. This is just inversion. You're just talking about the complementary inverted similar region. So even if it is red, it's showing that it's similar. Even if it is blue, it's still showing it's similar. All right. That whatever is here is found here. But if you're not seeing any line, it shows that that place is unique. All right. Yeah.
Thank you so much.
You're welcome. Okay, somebody is saying that how is different from BLAST tool. All right, see, BLAST tool, you can only use BLAST tool actually to some point in doing targeted genes. All right, and there's a perspective and approach to BLAST tool. All right, there's a perspective and approach to BLAST tool, because when you submit whole genome using BLAST, my God, the thing would maybe stay you stay there for quite a long time. It may not work. It may keep slowing because I don't think the database is kind of built for such a large numbers of genes. I mean, yeah, or genome. Okay.
Hello, thank you. One more question, please.
Yeah, sorry. Somebody said, can this software deal with rice genome? See, I told you either microbial, plant, or animal. Okay, so that's it. Go on, please.
One more question, please.
Go on, I'm with you.
Is there any difference between reference sequence and the query sequence?
Yes, reference sequence is the one you are referencing. All right, is the one you're referencing. It's the one you are focusing on. It could be your genome. It could be the main target. While the subject sequences are other strain. So if you are doing a PhD project and you have you did your DNA sequencing from the lab of a whole genome of Staphylococcus aureus, or maybe a plant or whatever, all right, your own is now the reference. While the other sequences or strain or species you get from NCBI database is now the subject sequence. Okay.
Okay, thank you. I'm talking about query sequence.
Okay, query.
If
query
reference
is
sequence
the same.
and query sequence are the
I
same.
get you. I get you. Now, query and reference, I would like to say is the same, because when you want to do BLAST, you will see query and subject sequence. Now, when you're doing BLAST, by the time you BLAST, the other sequence it brings for you as subject sequence, while your own is query. So query is similar to, is also referred to as reference, okay?
Thank you very much.
You're welcome. So I believe this is it. We have, can this software deal with human genome? See, yeah, it can, because the tools that are here, the tools that are in this particular software is built, is a kind of a general developed tool, all right, to do different kind of DNA of microorganisms. Okay, so you are good to go. Thank you, guys. And this software can do primary design as well.
Can the software used for cloning simulation?
Yeah, it can do cloning.
Oh,
you see, cloning.
Okay, okay.
So we have.
I have one question.
Sorry, we have a question here. How to identify and extract a gene portion in a sequence with primer sequence coding for this? I cannot show you with the region that code for the primer, but I can show you how to extract a specific reference sequence, okay? If you're asking of primer, you are asking, sorry, so you're asking, it's not part of this workshop. But if you want, you can enroll for a personal training session, one-on-one. So where we just show you how. Sorry, I'm looking for a unique region.
Hello.
Hello.
I have one question, please.
First of all, thank you so much for this informative webinar. I have a question. Can we do the same steps for gene comparison, not whole genome comparison? So we can trace the SNPs or the mutation in a single gene?
Yeah, definitely you can look for mutations, but it's not part of this session, all right? But you can do
the same steps instead of genome comparison. Can we use a single gene as the reference gene and compare it with different from different varieties or different species for the same gene, so we can treat the evolution of this gene? So the same step instead of the whole genome, do the same step for a single gene. This can be done or not?
Sorry, can you rephrase?
Okay, yeah. Can we follow the same steps to do a gene comparison?
Okay,
particular gene instead of whole genome.
Yeah, of course, you can.
Okay.
I have one question, please.
But see, wait. Hello. For you to do that, remember this is all genome. This is aligned all genome. So for you to do that, you do. I would advise you to do multiple alignments. All right, do multiple alignments. Is that clear?
Yeah. Okay.
If you're working on gene, a targeted gene, you can work with multiple sequence alignment. But if you're working with a lot of genes, you can still, if your genes is so much, you can still do whole genome. In as much as that gene has gene bank, all right? In as much as that gene has gene bank information. Okay?
I have one question,
so I can
sir.
extract the gene from, for example, 50 varieties and do a comparison between them.
Another thing you can do, another thing you can do is map to reference, but if you want to know how to do that, it's in the package that we're going to show you after this. Okay. It's in the
Sir, I have one question.
My question
Regarding this.
Ask your question.
is how can we align the context from the assembly data? Because when I tried, it gave me the error of the size, and when I tried to excuse me.
How can you do what? Can you ask the question again?
Excuse me, I can't hear you properly.
Ask the question. Ask the question again.
Yes, okay. I'm asking that how can we align the context from the assembly data? Because I tried, it gave me the error of size and asked me to go into the tools and to the preferences. But when I went there, it told me that I do not have rights to increase the maximum limit. So can you please guide me that if we want to align the context from the assembly data, how we will do that in this software?
Yeah. So please, if you want to do that, you need enough space for this work, for this software. Can you see mine is 3.396 MB? So make sure you install 64 bytes software. I mean, of Genome software, 64 bytes, not 32 bytes. Now, when you do, go to preferences.
Yes.
And at the preferences, go to general.
Yes.
Then you can increase the memory. But wait, listen very well. If your RAM is 4 gig, you cannot increase more than 3.400. If your RAM is 12 gig, you can increase to like 10 gig or 11 gig megabytes of available memory in the Genomes. So because the Genomes works with your RAM, so it means you need higher RAM in your system to be able to operate or work with higher size of genome. Is that clear? So when you do, you just need to click maximum available on Genomes and then you increase it. And when you increase it, you apply, you click OK, you close your Genome software and open it again. Is that clear?
Okay.
Thank you.
Thank you so much.
Thank you. Another question, please. Hello, the next person. All right, I believe there's no more question. So now we have a question on the chat room.
Yeah, excuse me.
Sorry, we have a question here. Good evening, Sandra Hazen. Sorry, I didn't call that. Firstly, Masters Bioinformatics, I have a question, sir. Does this software run on CPU or GPU? If this runs, okay, I think I've explained that just now. So just make sure you increase your RAM. Thank you. So we have.
Sir, I have one question. Yeah, can we please the software and for RAM?
For what?
Yeah, can we use this software and 4 GB RAM holding system?
I think you can try. You can try it if it works. Okay, it can work on Linux because the Linux version is available. I will send you the links. Okay, don't worry. I'll send you the links, guys. Please, if you go to Genome software to download this software, it will give you Genome Prime. This is Genome version 9.1.8. If you work with Genome Prime, we don't have the activation key for Prime. But the difference between this and Prime is just very little because of just from few updates. But all the plugin on Prime is also here. All right.
Excuse me. What about the position of this blue line? Is it significant?
It's very, very significant. I told you the blue or the red line is telling you the region of similarity. So whatever you have in the reference sequence, if you see a blue or a red line, shows that whatever gene is here is also present here.
Okay.
It's also present whenever you see. But if you don't see the red line like what you're seeing on my screen now, it's telling you that, oh, this particular gene is unique only to the reference sequence, but it's not present in other strains. Is that clear? Now, how do you extract this unique gene? Just right click on it and extract regions. Can you see? And click OK. And then it will show me. Now,
Sir,
we can see the region. Can you see the region? We just extract the region. You see? So you can save the sequence of this region. Go to Export, Batch Export, click Gene Bank Sequences, save it as the Gene Bank, and click OK. All right. By the time you do that, it's going to save, and you just go where you save it, you find it on your laptop, and then you'll be able to use it for whatever else you want to do.